Well, good afternoon, everyone. My name is Ray Kent. Welcome to our vaccine symposium on behalf of the Bloomsbury Set. I'm Director of Research Administration at the Royal Veterinary College, as you can see from the screen just now. And I'd like to welcome everyone to this symposium this afternoon. If I could have the first slide, please. And the next, please. Okay, thank you. So just by way of introduction to the symposium, I'd like to say a few words about the Bloomsbury Set. The Bloomsbury Set is a programme funded by Research England as part of the Connecting Capability Fund. It is a 3-year programme initially, which started on the 1st of April 2018. And we are a consortium of UK universities. We're all colleges of the University of London. And we are working together in a One Health approach, looking at the development of new vaccines, diagnostic tools, and mathematical models to combat infectious disease and antimicrobial resistance. And you can see the partner logos there in the slide. So the programme is led by the Royal Veterinary College. Partners are the London School of Economics and Political Science, London School of Hygiene, Tropical Medicine, and SOAS, University of London. And we work together as part of the London Interdisciplinary International Development Centre, LIDC. Next slide, please. So just to explain a little bit about the Bloomsbury Set, why are we different from your typical consortium? It is very much an interdisciplinary programme. We're trying to bring together the 3 elements here, science, economics, and technology in one place, hence SET. So it's not the usual definition of SET, but bringing together sciences and humanities, social sciences, to tackle some major problems around infectious disease and AMR. As a consortium, we have expertise in these different areas, and we're also well connected with pharmaceutical companies, with government agencies, research institutes, and so on, with a particular focus on working in low to middle income countries. The programme is not just about life sciences and the expertise there, but it's also about applying social sciences to some of these issues. And so working with policymakers and other key influencers in public health is a key aim of the programme. Now, to our knowledge, there's no other group within the UK that has been working in overlapping challenges in this way. So we are trying to develop diagnostics, support the development of new vaccines, and also improve our mathematical modelling of disease transmission and spread. And importantly, not just the science and technology, but to ground these in socioeconomic realities as part of a single interdisciplinary initiative. Next slide, please. So this might seem obvious to you coming to a vaccine symposium. Why are we focusing on vaccines today? I think when we proposed this programme just around 3 years ago, of course, we didn't anticipate the COVID-19 pandemic that has affected us all wherever we are, excuse me, wherever we are in the world. But it was apparent even then that infectious disease and resistance to antimicrobials used to control disease was going to be a major threat to human health. In fact, has been a threat to human health for many years. And in particular, looking at the rise of antimicrobial resistance in zoonotic pathogens, some of which are now leading to infections that are untreatable. We also knew and know that vaccines are costly to develop. They take many years, in most cases, to go through clinical trials. And even when they're ready, as we're seeing with the vaccines against SARS-CoV-2, take-up can be limited for various reasons, political, social, cultural. And of course, there are many lessons to be learned from this. So we've funded as a consortium a number of project grants and innovation fellowships in this area, details of which are going to be presented today, but with a focus today on vaccines, and further presentations will take place at our conference later this year. I'm going to introduce our first speaker then, without further ado. That is Dr. Phil Packer. Phil will be talking about vaccination research to commercialization, the funder's perspective. Phil is the innovations lead for antimicrobial resistance and vaccines at Innovate UK, which is part of the UK Research and Innovation. So I will now hand over to Phil to give the first presentation. Thank you.
Hello, everybody, and can you hear me all right? Thank you very much to the organisers for inviting me to speak. I'm going to move straight on after the introduction. And so if I could have the next slide, please. And this is a little bit of PR, but I hope that it will demonstrate how I fit into all this. Well, I work, as it's been said, for Innovate UK, which is part of UKRI. And essentially what we do is we take about £7 billion worth of government funding a year and invest it into research and innovation, funding both partners in industry and academia. And we're looking basically to create knowledge with impact. And next slide, please. And specifically talking about Innovate UK, our aim is to drive productivity and economic growth by supporting businesses to develop these new ideas. So essentially we connect businesses to the people that can help them, and we also fund businesses and research collaborations. And we do that right across the economic sector, different value chains, and in different regions of the UK. So if I can have the next slide, please. So bearing this in mind, I would just like to show you my very sketchy diagram of the vaccine developmental cycle, which I hope sort of reflects your view of it. And essentially it shows from the left to the right, going through preclinical and non-clinical developments into the clinic, and then it transfer into industry where you complete your manufacturing and your clinical program and go for a licensure. Now, traditionally, excuse me, most of the work on the left-hand side of this diagram is done in academia. And I'm going to say now that I think what the normal picture is a bit of an oversimplification, and to who performs what part of the developmental cycle. And I said traditionally the left-hand side is done by academia. So if I can have the next slide, please. And so that is where the likes of all the other research councils will fund work in this pathway. And on the next slide, you can see that as we move into industry and into the clinic, etc., we will see that we then get funding from Innovate UK. And then if you look at the next slide, what I'd like to say is I think we should have actually more integration between the two. And I think that we should have more industrial engagement in the left-hand side or the early development, and more academia involvement in the right-hand side. So without turning into Jonathan Van Tam, I'd like to use the analogy of building a house here. And I know that a painter and decorator probably understands and could put the roof on a new house that I might want to build. But if I want to have it done quicker and probably properly first time so it won't leak, I would actually prefer to have the roofer to do that piece of work. So if you look at the same analogy with when it comes to vaccine development, academics have their own specific areas of expertise, and industrialists or people in business have their expertise as well in their own areas. But they probably all understand the process of development of a vaccine. Excuse me. And so I'm not actually criticizing anybody in that point. Everyone's got their own level of expertise. But if I take the example of clinical trials, I believe that they should be designed, organized, and run by a clinical research organization. And this will increase the likelihood of them being completed effectively, quickly and safely. Therefore, if an academic organization, for example, gets a grant and they've got a clinical trial in their program, I believe that this work should be contracted out to a clinical research organization, or better still, I think that organization should be integrated into the team, because I believe that that multidisciplinary team is more likely to succeed. And if you look at that picture a bit bigger, and give some other examples quickly while I can, I think if you bring your academic into the early movement into industry of a product, the industry will have the expertise to see that when they start to develop a process in manufacturing, any slight tweaks to a molecule, they can then talk to the academic and say, is this going to affect the efficacy, etc., rather than going back when it doesn't work later on in time. So I know I've now got to move on. So I'd like to see the next slide, please, and talk a little bit about where most of the work is funded for vaccine development in the UK for man, and that comes through the vaccine network funded by DHSC. And this is to support the government to identify and shortlist target investment opportunities for the most promising vaccines and vaccine technologies to help combat infectious disease with epidemic potential. And that is something that is particularly important at the moment with COVID. So if we go on to the next slide, what I'd like to show is that a lot of that work, which is directed mainly at low middle income countries, is actually delivered by by UKRI. Now, I can't go through all these different bits of work that are going on, but I hope you can see there's a good plethora of work going on in these areas done by the different organizations within UKRI. And on the bottom of the slide, you can see the bit of work that I'm responsible for, which is the delivery of about originally 49 vaccine projects to develop vaccines to the preclinical and into the developmental phase. Can I have the next slide, please? So very briefly, we also do work ourselves within UKRI, and you can see here, I can't go into all the details of all the different bits of work that are going on in the research councils, and you can see that most of that is on the left-hand side of the diagram. On the next slide, you can see some work that is going on in Innovate UK, and you can see, hopefully that comes up, yeah, you can see that we fund more to the center and more to the right, and we develop different stages of the clinical pipeline through the biomedical catalyst, and this is for human work. And there are different sums of money in these different stages. And you can also apply to do human and veterinary vaccine development through the grant, through a smart grant, which you can get for up to £2 million for. Now, I'm conscious how much time I've got, so I'd just like to go on to the next slide quickly and point out one thing I have missed off is that, and something's not come up, don't worry, is that we also fund the vaccine Manufacturing Innovation Center, which will help the development of these vaccines going forward. But the last thing I'd like to point out before I conclude is that funding of these late stage clinical development is traditionally funded by industry. But for diseases with epidemic potential, they're not attractive for business because there is no long-term guarantee requirement for a vaccine supply. So that's an issue that we have to bear in mind. So if I can have the next slide, I'd like to just bring all this together and say we've done a lot of work over the last few years, and a lot of this has been very helpful for development of the COVID response in the UK. So through the vaccine programs, the two vaccines which have been mostly funded now in the UK are the Oxford and the Imperial vaccine, and they came out from platform technology developed in some of the projects that we've been funding and delivering as part of the vaccine network, which is a good thing. And those two vaccines have then been taken further and into clinical trials and phase 1 clinical trials. And as you know, and that's been done by MRC, that the Oxford vaccine is now licensed. And we've also taken some of those programs, 7 in fact, to repurpose them to develop future generation COVID vaccines. And Innovate UK also have got their own program where we've funded 5 vaccines in this area. But importantly, I'd just like to point out that 2 of these have been picked up from the repurposing exercise. So you can see how all the work that's been funded in the past has been taken forward and is helping the COVID pandemic. And I will now finish there, and just to say we are in contact with the FAST task force. Okay, thanks very much.
Okay, thank you, Phil. That was really interesting talk, and I think probably I hope you're able to save your questions for later on in the session, but please think about questions that you might want to ask any of our speakers today, and have that opportunity a little bit later on this afternoon. Okay, it's my great pleasure to introduce then our next speaker, Professor Brendan Wren. Brendan is Professor of Microbial Pathogenesis, and he's also Dean of the Faculty of Infectious and Tropical Medicine, Tropical Disease Medicine at London School of Hygiene and Tropical Medicine. And Brendan is going to be speaking to us about development of low-cost glycoconjugate vaccines for pigs. So over to you, Brendan. Thank you.
Okay, thanks, Ray. On the first slide, please. Yes, so first of all, I'd like to say good day to everybody, wherever you are in the world. And last year was a terrible year as mentioned, for the COVID pandemic. But a bright point is the vaccine. So we have had new vaccine technology platforms available, and I believe it's a golden era for vaccinology. And I'd like to take this opportunity to share with you a vaccine technology that we developed over the last few years. So on the next slide, so this slide shows glycoconjugate vaccines, which is the area that we've been working on. These are highly successful vaccines. They've been used for humans. Over 1 billion doses have been given. The advantage of them is that you get T-cell dependent and independent response. And this means that they're highly effective, they have long-lasting memory, and they're suitable for children and adults. So as I mentioned, billions of doses have been given to humans, but to date, ironically, they haven't been given to animals. So humans has been the guinea pigs here for animals. So on the next slide, this shows the method that we use, which is simple biological conjugation. So the reason why vaccines have not been used for animals, glycoconjugate vaccines, is the cost. So you can see here at the bottom of the slide that the chemical process is highly complex. There are over 300 steps for purification. So our approach is to persuade a bacterial cell to make this glycoconjugate vaccine, which consists of a protein of choice, a glycan, that could be a capsule or an O antigen, and then we have this coupling enzyme, a magic coupling enzyme, which will stick the glycan to the protein to make the glycoconjugate vaccine in a one-step process. So on the next slide, we show how we do this process now. So it's a rather simple process. Nowadays, we can DNA synthesize the protein of interest and put in so-called glycotags. Also DNA synthesize or clone the glycan of interest. And then we add those to E. coli cells that contain the coupling enzyme. And then, as I mentioned, it's a simple case of growing up the E. coli and producing large quantities of the glycoconjugate vaccine. So on the next slide, it shows the areas where we've done this. So the big advantage of a technology, apart from cost, is the flexibility. So we could mix any particular protein of interest with any particular glycan in most instances. So the areas where we've done this is in pre-existing vaccines. So for example, the pneumococcal vaccine, and I'll show one example of that. The other areas where the new vaccines are required, and actually for most bacterial diseases, there are no vaccines. So we've been working on at least a dozen new vaccines in this area. And then finally, the other area is new markets. So this is potentially for livestock and for animals. So on the next slide, I'll show an example of the technology. So the green banding here is basically the result of the coupling of the glycan to a given protein. On the left, this is a protein called ACRA, which is originally from Campylobacter, the enzyme where we discovered the bacteria where we discovered the enzyme. And then in the middle panel there, you can see that we call this a second generation vaccines, whereby the banding there is examples of 3 protein candidates that we've made, and we tested these in the animal models, and are at least more effective than Prevnar, which is the current industry standard that's been used worldwide. So we've established real proof of principle for pneumococcal vaccines. So moving on to the next slide. So these are examples where I've used it for veterinary vaccines. So for example, E. coli and Salmonella are attenuated strains that are used in the poultry industry. So we've essentially piggybacked on a Campylobacter glycan coupled to a Clostridium perfringens toxin to make a triple vaccine. And these are currently being tested in trials with an industrial partner. The second area here is in the poultry, the pig industry, which is relevant to the Bloomsbury SET proposal. So here we are looking at Streptococcus suis and Actinobacillus pleuropneumoniae, and we have other examples there, such as Coxiella, Brucella, and many other animal diseases. And more recently, we've spun out a company, ArcVax, which has started at the Babraham campus just this year, just a few weeks ago. So on the next slide, so it shows Streptococcus suis, which is a huge problem for the pig industry, and it causes much deaths, particularly in young pigs. It's also a zoonotic disease, and I remember many years ago when I went to Vietnam, a young clinician called Jeremy Farrar showed me a ward full of patients, and it was the leading cause of septicemia in Vietnam, or certainly in the wards in Ho Chi Minh City. So, so it's a considerable problem, not just for the pig industry, but for humans as well. So on the next slide is the technology that we developed for Streptococcus suis. So this is called a one-pot proof of principle that Liz in my group has really elegantly developed. And it shows an example where we can mix the components. So that's the capsule from Streptococcus suis to a given protein, along with the magic coupling enzyme, and the green bands there demonstrate that we've established proof of principle for doing this. So on the next slide, the second part of what we do is that we'd like to make this a dual vaccine, not just against Streptococcus suis, but for Actinobacillus pleuropneumoniae, which is another huge problem in the pig industry. And with the classic vaccine candidate, the toxin hill, we've made many constructs, and we've copied in the appropriate glycan sequon, and we've demonstrated that we can actually glycosylate the Actinobacillus protein carrier. So this all looks promising, and we hope to carry this on throughout the Bloomsbury SET project. And next slide. So just to summarize the advantages of the method, essentially we're cloning the vaccines in a one-step process. So this means that it is low cost, has real flexibility in design, that we can start to couple different combinations of proteins and different glycans. I mentioned about the concept of a double hit vaccine, where in fact the carrier protein and the glycan can be from the same organism, or they can be from different organisms. And finally, just to reiterate the concept that for veterinary vaccines, they're a bit in the dark ages. Some of the technology is really basic, going back many years. Now, I hope that this changes, because improved vaccines for veterinary vaccines doesn't just mean economic prosperity and health for the animals, but certainly for zoonotic diseases has huge benefits for human health as well. And not least, better human and animal vaccines will mean less antibiotic use. So there's a real drive towards improving veterinary vaccines. And on the final slide, just to acknowledge that part of the grant, or the Bloomsbury SET grant, is with Dirk Werling and his colleagues at the Royal Veterinary College. And I acknowledge John Kukui, Ian Passmore, and Lizzie Atkins, who works in my group. Dirk's role in this being the veterinary side, he's done an excellent job in terms of producing a nice model of the disease. So I'll finish there and take questions later. Thank you.
Okay, thank you very much, Brendan. Our next speaker is Dr. Virginia Marugan Hernandez, who is a research fellow in vaccinology at the Royal Veterinary College. And Virginia is going to be talking to us about poultry vaccine platforms for use against transgenic Coccidia parasites. So over to you, Virginia. Thank you.
Good afternoon, and well, I want to thank all the Bloomsbury Set team to organize this event. And well, if we can start with the presentation, please. And yeah, well, my name is Virginia, and I'm gonna introduce our Bloomsbury Set project. Today, that's entitled Development of a Poultry Vaccine Platform based on transgenic Coccidia parasites and evaluation of immunoprotection against a necrotic enteritis. Next slide, please. So, well, that's common symptom. You cannot see very good the graphs, but it's just to show how the poultry industry is in continuous development. It's constant growth from the last 20 or 30 years. And every system, intensive system or backyard systems, are under the threat of many different infectious diseases. Next slide. And also, as you can see in the back, is or especially chickens are under very intensive vaccination programs. And that's the reason why we suggest using Eimeria parasites as a vaccine vector. Next slide. Very briefly, I want to introduce you Eimeria. So you can see here in this tree is there on purple at the bottom, and it's a Epicomplexar parasite closely related to many other parasites that are very important in humans and animals. Eimeria parasites cause coccidiosis. That is an enteric disease that has a direct impact in production in many animals. In the case of chickens, the cost of the disease and consequent treatment or prophylaxis are estimated in over 10 billion per annum worldwide. And the control is mostly achieved by chemoprophylaxis or live vaccination. Next slide. So why we think Eimeria could be an interesting vaccine vector? So it's host specific. That means it will be fairly safe for any other species, and it's very immunogenic. So it could be a very good vector to raise different types of immune responses. Can be also administered orally. That is really interesting in the case of chickens, and has a large genome. So we could probably accommodate genes for several pathogens at the same time. And as I mentioned, there is a commercial live vaccine for coccidiosis. So if we could incorporate more pathogens that could be vaccinated on the same system, that would definitely add value to this vaccine. And in the past few years, there have been a lot of improvement using these transgenesis technologies to express antigens in Eimeria. It's well standardized for different species and has been used in basic research. And we have some good results when used as a vaccine vector, but we still have some limitations, as is the random integration of the transgenes, the difficulties to stabilize our populations, and also with the levels and location of the transgene expression. Next slide. So we decided to move forward with this, and we put a project to Bloomsbury Set in partnership with the London School of Hygiene and Tropical Medicine and MSD Animal Health, with the aim of develop a platform for easy exchange of antigens, so we could change for different pathogens in an easy way, incorporating the CRISPR-Cas9 technology in these parasites. We will also aim to improve the expression and the exposure of our antigens to the host, to probably hopefully improve the immune responses. And we choose to target on first place other pathogens that cause disease on the same area as Eimeria, so enteric pathogens, starting with Clostridium perfringens. Clostridium causes necrotic enteritis that is being controlled through antibiotics, but all the new regulations in banning antibiotics and inophores is making a re-emerging disease, at least in the States at the moment. The next slide, please. So that's not animation, but so the first at the bottom, that was our initial plan, was just to start with all the different objectives one after the other. So we will generate all the elements that we need to standardize the CRISPR-Cas9 and the parasites, generate our editable strains on the top of that, prepare our construct for the antigen expression, and then generate our vaccine strains that they will be characterized for the gene insertion and levels of expression, immunogenicity, and protection. But we have to, in the top, we have to actually start overlapping objective. Next slide. So in first place, we adapted CRISPR-Cas9 technology using some plasmids from Toxoplasma. So we have to standardize and add our own promoters. And I'm not going to go in detail, but we did a proof of principle in which we managed to knock out fluorescent on a previous fluorescent population that proves that our elements were already in place. I want to highlight that we don't have an in vitro system for Emeria parasite. It has very complex life cycles, so we need to rely on animals. So we generate our parasites on the chickens, and we use fluorescent markers for their selection by fax. Next slide. So in a similar way, Emeria relies on random integration, or the genes are integrated randomly. So we decide to knock out one of the proteins that are involved in this mechanism of random integration. In that way, Emeria will need to use homologous recombination to integrate genes. So we modify slightly our plasmid to target this new protein, and we generate the knockouts, and then add in some fluorescent markers to be able to select our parasites. And this is something we have already generated some population, and we are in the process of validate them as knockouts and see that the parasite are integrating biohomologous recombination. Next slide. So this part was going to be done after generate those parasites, but because of the COVID, we start with this work after summer instead to wait to have these parasites. So what we did is using our select Clostridium antigens, we add some regulatory elements that we have seen before that they can produce secretion or expression on the surface of the parasite. And we will link those to a fluorescent reporting to allow us to select the population. But they will be expressed in a way that the reporter will not interfere much with the folding of the protein, we hope. So next slide. So in a similar way, we generate our construct, but then on a wild type background, not in our editable parasites. We have transfect this new construct, put them on the chickens, and we have been selecting them. And we are obtaining our populations that we hope to be able to characterize soon in January, February, to see that they're actually expressing the transgenes at the levels we would expect, and that the infectivity hasn't been compromised. Next slide, please. So once we have these parasites ready, hopefully by March, we could start some immunogenic trials in which we can use the parasites with protocols we already have standardized and try to see different types of humoral and cellular immune responses. And if we get some positive results, then we will move to vaccination trials, again with models that have been already standardized for Clostridium perfringens, and see if what we manage to get the results we expect. Next slide. And I just want to thank everybody in the audience and to everyone involved in this project and the different teams at RBC, the London School of Hygiene and Tropical Medicine, and MSD Animal Health, And if you have any question, I will be glad to answer them later on the Q&A session. And yeah, thanks again, everyone.
Okay, thank you very much, Virginia, for that really interesting talk. Our fourth talk in the set this afternoon is from Professor Richard White and Puck Pelzer. Richard is Professor of Infectious Disease Modelling at London School of Hygiene and Tropical Medicine. Puck is an epidemiologist working on TB, and their talk is going to be on multidisciplinary research for the strategic and translational development of late-stage tuberculosis vaccine candidates. So over to you, Richard. Thank you.
Thanks very much to you, and thanks very much to the organisers for inviting us here to talk today. Our talk is going to be very much at the other end of the development process. We've heard some very, very basic science, some fantastic basic science talks. But our talk is very much about, okay, so you've got a vaccine, what are you going to do with it? And in particular, how are you going to maximize its use, its benefit to the population you want to benefit, and how you're going to scale it up and implement that vaccine, which is, Phil was mentioning right at the beginning, actually is quite a hurdle and often overlooked. So this work is part of a wider cooperation with the London School of Economics and indeed KNCV, Puck here today, and also our country colleagues in India and China, India and South Africa. Okay, next slide. These are TB vaccines. TB is bad. It infects about, or that's about 10 million cases of TB a year. And unfortunately, about 1.5 million of those individuals die. And we are getting on top of TB, but incredibly slowly. The phenomenal rate of decline of about 2% per year. So other tools and approaches are certainly necessary. Next slide. And there is a plan. WHO has a plan, and you can see the graph of how we're going to get on top of TB there. Basically, into the first 5 years of the kind of first 10 years of that, which is to scale up new technology. And to be frank, that's not going so well. So we're still up at that 1.5 or 2% trend at the top, rather than that rather impressive slope going down. But there's plenty of room for improved new tools like TB vaccines, and that's kind of in this graph, in this cartoon, expected to kick in around 2025 at the end of the development process. Next slide. Fortunately, people realized good 10 years ago and 20 years ago that TB was not going anywhere fast, despite our new drugs, and they started investing in TB vaccine candidates, and there's quite a few in the pipeline, as you can see right there. Different indications, infants and neonates there at the top, adults in the middle, and therapeutic vaccines at the bottom. Different stages of the clinical pathway across the top there. I'm going to draw your attention to the one that's kind of in the top of the adults and adolescents, which is M72, which is a vaccine that's just had some very positive results from phase 2b trials, and we were focusing on in this project. Next slide. M72 demonstrated about a 50% reduction in efficacy against TB disease, the thing that kills people, in a multi-center phase 2b study in already infected individuals in South Africa, Kenya, and Zambia. And that leads funders, fortunately, that was enough to get funders thinking about how we might take this into phase 3, and if so, how we might implement it if it comes out of phase 3 successfully. Next slide. Our group uses models to do that, and we see kind of model as a kind of logical framework where you can set up a model of a new vaccine, you can implement it in different populations, and you see what you get, and you make estimates for how much it might cost you. In essence, it's that simple. Next slide. We already have done that to M72 in some very simple caricature introduction scenarios, which typically assume that you can scale up your vaccine instantly at the January the 1st of a year to about 70 or 80% coverage. Now, that's very kind of simple, and that you get results like this if you do so. but that leaves big gaps. That's never going to happen in countries, and we don't know how countries are going to use this vaccine. So, next slide. This project is all around the bullet, looking at the social science research, talking to people in countries about how they might use this vaccine, and then reprogramming those models so that they better reflect how they'll actually be used. And that information can be used to feed back to those decision makers to think, well, actually, I can't afford to do that, but I could afford to do this. And if I do that, actually, it doesn't really do very much. But if I do that, do this other thing, I might actually get a good bang for my buck. So that research is basically what we're doing here. Next slide. We were going to do that in South Africa and India, but unfortunately COVID got the better of us, like other people. So that research is just now, the interviews happened in South Africa, but not India. So we're just taking it forward in South Africa. Next slide. And it's basically to do those interviews, to reprogram the models, improve the models, better modeling tools, improve them, apply them to the character of population in South Africa, work with the decision makers to say, okay, this is what you're going to get, and then help inform their use in country when it becomes available, but also feed that back to global decision makers. Next slide. I think this is where Puck is going to tell us about the cutting edge, very recent social science results from South Africa.
Right, yeah, so we conducted individual interviews with several stakeholders in the countries, and they aim to represent different aspects of for the vaccines. The interviews consisted on different sections. We had a section on acceptability and feasibility, delivery of TB vaccine, and any other previous experiences on other vaccines. We analyzed the qualitative data via thematic analysis, and the implementation scenarios and costs were summarized in a tabular form. Next slide, please. So some preliminary results from South Africa is that overall stakeholders had a very strong acceptability interest in a new TB vaccine for adolescents and adults. This because of the very high TB prevalence and HIV prevalence. The vast majority was confident that we could start targeting within the next 2 years, depending on logistic constraints and approval. Some challenges that were named was that M72, the new TB vaccine, requires a second dose, which could decrease the coverage. BCG vaccination, which is another strategy opted, was considered less likely, as this can give some very nasty side effects in people living with HIV. Also, COVID vaccination strategies can inform TB vaccination strategies, so that could be a benefit for TB introductions. Next slide, please. And here you very smallly see an example of an implementation strategy. Several populations were named by the stakeholders. The most often named was adolescents, as we could reach these people through the school-based vaccinations program, which is already in place, which is a facilitator for this type of strategy. Some challenges that could be faced is that there's stigma to TB and TB vaccines and logistical constraints of implementing a new TB vaccine. Regards of costs, most stakeholders were willing to spend the same as HPV. Next slide, please. So some preliminary conclusions is that acceptability overall was high in South Africa, and that our study provides some context-specific challenges to the implementations of TB vaccines targeted to adolescents and adults, which can inform development strategies, policy, and practice. Back to Richard.
Thank you, Puck. And they've literally come in at the end of last year, so very exciting. And this week, we're starting to model those results, and we should be informed, talking to the funders or decision makers in South Africa kind of middle of next month. Next slide. I'll be super quick. So although the actual work has been delayed by COVID in country, we've managed to get very closely engaged with the key decision makers in the field, particularly WHO, who we've talked to them about the M72 vaccine pipeline, the clinical development pathway, and the licensure strategy. The HIV network is now interested, has now allocated funding to TB vaccine, and we're very keyed into that. And indeed, the clinical trials vaccine consortium for TB vaccine discovery have recognized this end stage decision making. Now we have these positive results. Our phase 2b trials are critical, and they set up a whole research community and invited one of us to be co-chair of that. And indeed, it's led on to future funding from the foundation and in WHO, who recognize the criticality of this sort of work. So thanks very much to SET for this initial seed grant, and we hope to have more positive results by the end of the grant, which will be end of spring this year. Over to you, Ray.
Okay, that's great. Thank you, Richard, and thank you, Puck, for that interesting presentation. For those of you out there listening to this now, could you start thinking about putting your questions into the live Q&A box, which you should see on the right-hand side of your screen? We're going to move now into a panel discussion session, and we'll be taking questions from the Q&A box, so please start to put those in. The panel discussion is going to be facilitated by my colleague, Professor Dirk Whirling. Dirk, with that nice snowy scene behind him, is the Professor of Molecular Immunology at the Royal Veterinary College, and he's going to lead the panel discussion, but you can put your questions to any of the speakers from this afternoon's session, or indeed to multiple people, depending on your interests. So over to you, Dirk, to chair this panel session. Thank you.
Okay, thank you very much for all the presenters to staying on time and to provide us with some really exciting data. I mean, we had a quite broad area we covered, from glycoconjugated vaccines for pigs to transgenic coccidia to uptake of vaccine by stakeholder. I hope there are questions coming from the audience, but maybe I could ask a general question to all the presenters. Now, we see that within the vaccine design area, we are developing more and more DIVA-based vaccines, which are all based on some sort of genetically modified research. Do you think, and this is an also question for Richard, do you think that this will positively or negatively potentially impact on how these vaccines are taken up by stakeholder, being either the ones we are injecting with, farmers, or the society in general?
In terms of kind of vaccine confidence, Phil?
Yeah, because we always have that genetically modified is not good approach. Yeah. However, if you think about specifically veterinary vaccines, these veterinary vaccines at the moment, at least, are you take your pathogen, you either kill it and have an inactivated vaccine, or you have a modified life vaccine by attenuating this pathogen. And we have no idea how these vaccines work, whether this is really the best approach we have. However, as soon as you say to the companies or to other stakeholders, well, we have actually a better vaccine approach, however, that involves a genetically modified organism, then we get this idea of, oh no, we don't want to go there. Now, do all of you think that this is only an issue that we need to convince the law people, the legislative people, that this is actually not a bad thing because we can differentiate then between vaccinated and infected, or is there more behind it? Do we need to have a complete change in our society in accepting that these new vaccines may be actually far better?
I think that's a great question, Dirk, and apologies for calling you Phil. And I'm certainly not an expert in there. There are many people.
No,
sorry. I just want to finish my point. I'm not an expert there, and there are many people. I just want to flag up the Vaccine Confidence Group in the London School who would give you a much better answer than I. But my initial reaction is that there are parallels there, there's analogues there, aren't there? Because I think people had similar concerns around the rollout of HPV, which was basically designed for vaccine for young girls to prevent them against a sexually transmitted disease, and could have gone awfully wrong had there been hadn't been a really strong messaging campaign which effectively, as I understand it, and I mean, again, I'm no expert in this area, message it around a vaccine for cancer and basically downplayed the sexual transmission route of the infection in that whole messaging campaign. And if they can do it for something as emotive as sex in young girls, it doesn't seem beyond the wit of people to message genetically modified vaccines in a suitably sensible way to minimize the likelihood that it won't, the acceptance will, well, maximize the acceptance.
Yeah. So do you think that, Richard, in that context, do you think that it has also potentially to do with the fact that as this is a positive news, it is not picked up by the daily newspapers because it's something positive, and therefore they can't sell it because it's no scaremongering process in there? So does it mean that us as the guys who are developing these things need to find outlets where we can actually provide the lay society with a far better insight into, do you know what, these vaccines are actually done and tested, they work, however, no one reports about it because they work?
I think that's a great point. And I wouldn't be surprised. I mean, my exposure of this sort of kind of clever coordinated strategy is through organizations such as the Science Media Council, which was set up around, as you know, around the autism debacle, and positively works with reporters on, for example, in the UK, positively works with reporters on the major, or where they can, on the major papers, so that they gives them a route to well-informed scientists when these stories come along. But I mean, I've talked to the HPV people, because it sounds like they did a brilliant job. Not
Well, yeah,
expertise.
Yeah. As there are no further other question coming in so far, I'm in a very nice position that I can ask you all the question I have. Brendan, I got a question for you. I really like this approach of glycoconjugated vaccines, but how difficult is it actually to identify what kind of carbohydrate structure is a suitable antigen?
Well, I guess we're going on decades of data from human immunology, whereby glycans, which invariably are on the surface of bacterial cells, whether they be capsules or O antigens, do make good vaccine candidates. As I mentioned, billions of doses are given. They could also be glycoproteins as well. So there are three different sets of glycans that we look at, but because they're on the surface, because we know that they intimately link in with the immune system, they are invariably good vaccine candidates. And particularly when they're coupled to a protein, you get all the advantages of long-lasting memory, etc. So as for choosing, what is the best glycan to use, I mentioned about strep pneumonia. Obviously, we went for the capsules because that's proven. That in that case, we're trying to make the vaccines far more cheaply. Some of what we choose is ease, because it's a little bit of a complicated process to clone a glycan. So some of them are easier to clone than others. Some of them are not on an operon, so that make it a bit more tricky. And we've come up with ways to overcome that. So it's a bit horses for courses. I mean, one area that I've not touched on at all is the Mycobacterium, because it's far too complicated for me, Richard. It has a really complicated glycostructure, which is why it's stained differently, and they grow too slowly. So I've never really gone the TB route, but because I'm just a bit too impatient. So to some extent, we picked the lower hanging fruit. And following up from your previous question, I would say that in the veterinary industry, there are a lot of attenuated strains that are used in vaccines, and not least the ones I mentioned from Zoetis, the Polvax strains, the avian pathogenic E. coli, and the Salmonella that is being used, for billions of chicks and poultry worldwide. I guess that's kind of gone under the radar a bit compared with a human vaccine that is genetically modified. But I think it's our technology actually would only piggyback on an existing attenuated strain that is licensed. Most of the vaccines, including the pig one, we produce an E. coli, but that is a separate product, so it's not actually a live. It's not actually a mutated strain that's used at all. But it is about education, and I've been in this game for a long period of time. It seems to become more and more acceptable that there have been little issues with genetically modified strains, all the fears about them in the environment, etc. And as I said, given the billions of doses of poultry vaccines are out there that are essentially genetically modified in a knowledgeable manner, I should say, then maybe it's not as big an issue as we think. But I think involving these on the human side would need my colleagues at the London School that are vaccine confidence people, etc., to really put out a clear message.
Okay, thank you. Virginia, there's a question for you. So it's a very interesting approach to modify a parasite to overexpress an antigen to provide immunity for another disease. Has this been used in other systems as well? So the person who's asking that question is specifically thinking about bovine TB.
Okay. Well, I quite focus on now this chicken area, but they actually area parasites infecting bovine. So something that hasn't really been done. And I think I'm quite focused in this side, like thinking on that options using other parasites or Discoccidia in this way. But I think, well, because all these technologies are quite behind other pathogens. And I think there's much more going on in chickens. It's been more bested. We're using Meriatinella. So something, I think that's the limitation. Like basically, we don't have the tools, but as soon as we start adapting some of the parasite, it might not be too difficult to transfer. And I think it's something that might eventually, if we successful or a bit successful in this side, I think is a good option because we have Imerias infecting almost every species, no human.
Yeah.
So yeah, I think it's good.
I guess it's similar to potentially some of the glycoconstructs Brendan was talking about. Once you have a platform and you know how to do it, then expanding that becomes hopefully slightly easier. Pak, there was a follow-up from you on the question I raised.
Yes, on the genetic modification of vaccines. Yeah, I just wanted to add to that I think what we've noticed while conducting these interviews is that this genetic modification, for example, could be one of the many barriers that could keep a person from getting vaccinated, but it's very context specific. In one country, this could be very important, but in another country, it would be very important that a vaccine is produced halal or that a vaccine is given orally. So I think it's important, but also very context specific.
Okay, thank you. Can I remind the audience, please, that if you have questions, put them into your chat box on the right-hand side of the screen. And there is a question from Richard to Phil.
Yeah, Phil, I love your introduction. It was very positive. But you did raise some big questions in there, one of which that stuck out, which is an issue, I think those of us working in vaccines for low and middle income countries feel very keenly, which is you said that industry does vaccines for rich countries and doesn't do them for poor ones, basically. But you obviously didn't go into what you thought about that and what you think we could do about that. I wondered if you might want to elaborate.
Yeah, and I think, can you hear me all right? I think it is a
bit louder, please, Phil.
Can you hear me now? Is that better? Yeah, I think it is a problem, and I don't know the answer, but I think so. I think at the moment what we need to do is get enough vaccines against enough diseases up to the fact that we've got them in the clinic. They've been proof of principle, and at least we have them there ready in case something does happen, and then they will be picked up. And I think the COVID example is classic, where when it came down to it, government have thrown a lot of money at this, but that is still going to be a problem in low and middle income countries, potentially because they haven't got the money. One thing I think we might need to do is, so I do a lot of work on the AMR side, and we're looking at pull initiatives whereby government are trying to find mechanisms where they provide a manufacturer or a pharmaceutical company money to allow them to produce, in that case, an antimicrobial, but in this case, it would be a vaccine, which is actually not used. So you have a stock of it there ready. Maybe you wouldn't have enough for a worldwide pandemic, but just initially for enough if there was a big outbreak, and essentially they're put on the shelf, and they're only used if they're needed. And the company, but that is a big investment. We're just seeing that happening in AMR. People beginning to think of that solution, but it is expensive. But I think it might be the only sort of solution in the short term. Because these are businesses essentially, yeah. and they've got to try and make money.
Yeah, there's an actually very fitting follow-on question from the audience to that, whether all of you doing vaccine design and trying to assess stakeholders' opinion or what are the problems in the vaccine chain, so to speak, whether we take into account possible constraints of formulations, technical, cost-wise, logistic, on production and upscaling, as well as further issues such as storage, cold chain issues in LMICs. And how would we overcome that? And so this is, I guess, a question from the audience to all of you. So if you want to comment on that, just wave with your hand and I will pick you out. Phil, let's start with you.
So I think the COVID example I can use, and initially, of course, the first vaccine, which was licensed, had all sorts of problems with the cold chain. Well, it's not a problem, it needs a big cold chain, and that is not going to be possible across the world. And I think there is an incentive on government to be persuaded to invest in future vaccines or next generation vaccines, even when we've got one ready, so that it can be used, has a better mechanism of use. So if it can be, say, needle free, or it can be stored at room temperature. So I think we've got to encourage that we don't just have one on the shelf and think that's the answer. I think we've got to keep working to find new solutions. Not easy when money is tight.
Not easy when money's tight. And just before someone else potentially chips in there as well with an answer, there's a comment in the audience saying that I'm concerned about the implication that veterinary vaccines are not as advanced as human vaccines. Many of the latest vaccine platform technologies have found their first application within animal health, and there are also many novel adjuvants and delivery system used. Genetically modified vaccines are generally well accepted within veterinary medicine, and the regulators support the licensing. So is there, if you take this into account, are we just suffering from not enough communication between human medicine developers and veterinary vaccine developers, which is interesting because if you think about the companies involved, Boehringer, Zoetis, Pfizer, AstraZeneca, they more or less either had all once a veterinary site or still have them. So how can we impact on that? I mean, this is what Bloomsbury said in Partial is about. Brendan.
Yes, well, actually, the real driving force for us in terms of producing veterinary vaccines is that they're quicker to get to market. The regulation hurdles are less, and that's why after at least a decade trying to do this on the human side, we spun out the company ArtVax, which will be dedicated towards veterinary vaccines. It's true there are a lot of innovations that are tried through the veterinary field, but actually, if you look at most of the vaccines, a lot of them are dead strains, as you'll know, Dirk. There's a real mixture. I agree that there's some innovations as well, and it is good to try innovations on the veterinary side, as I mentioned, where regulation is less and there are less hurdles. And the other huge advantage of actually going into the veterinary side is, for many years, being on the human side, making many different vaccines that are perfect for mice, getting them to humans is at least we can go straight to the actual species. So, and I did allude to in a talk, I think the other social advantages for veterinary vaccines do link with humans in terms of zoonosis and less antibiotics in the food chain. And Virginia mentioned about Clostridium perfringens and ectrizing enteritis is a perfect example where in the EU we use less antibiotics, were not used for farms, and then suddenly this organism became really prevalent and caused a nasty disease. So we do have to be smart and develop vaccines. So I think so yeah, that is the real driving force for us, is to go to the veterinary side over the last few years. So that we can establish many proof of principles, including through the Bloomsbury set, and this will give us a lead later on into the human market.
Okay, can I follow that up with a hint or a remark to Phil and UKRI Innovate network? I think the point with the mouse work is really a very important one, because us working on the veterinary side, we have seen that vaccines that work very well in mice do not work in the target species. But if you turn this around, you could also argue that a lot of vaccines that did not work in mice could have potentially worked in a target species. So what it comes down to is that I don't know how. Probably we need to plant more trees that can grow money on, but theoretically it would be really nice if we can do more real experiments with the right host rather than going the mouse route.
Yeah.
I know. This is something we can probably discuss over several pints and don't find a solution. But I think it's a real issue that we do a lot in mice where we know we can bias the immune response to a certain extent by picking a mouse model, and then it has no relevance whatsoever.
Yeah.
For the real world, specifically as stick with COVID, we have no challenge model in mice.
Yeah. Yes, it's an interesting point, and I'm not going to try and answer it off the top of my head, but I think it is. But I think you've hit a good point there, and how we can look at that and try and think about that in the future. So that might be an innovative approach to try and avoid that failure issue. Yeah, that's a good question. So I've written that down. Thank you. It's something
most
to think
welcome.
Yeah.
There's again not really more question coming in from the audience. There's a comment which goes back to a point Phil made. Pull mechanisms can make a difference to facilitate early uptake in niche markets. I see the same in human health with PPC and PPI initiatives, so innovative procurement from the European Commission. I guess that's something we can all think about, and actually where we could learn a lot from each other. I know that one of the most driving force in veterinary medicine is cheapness of vaccine and mouth to mouth. So if one farmer uses it, the veterinarian goes to the next farmer and says, oh, your colleague down the road is using it, seem to have good experience with that, how about it? And then they can buy bulk. So yeah, I think it's not only working for human medicine, it's something we can pick up on in veterinary medicine as well. We have about 10 minutes left for questions. Is there anything else coming from the audience? Then I have a last question maybe to Richard and Pak. So I was a bit astonished really to see that you say that the acceptance for vaccination in South Africa, for TB vaccination in South Africa, seems to be quite high. So what then is the major obstacles to get the vaccines actually out there? Is that the costs of the vaccine, so that the Joe Blocks on the road, on the street cannot afford the vaccine?
Well, I mean, the block right now is that we haven't had the phase 3 trial that's confirmed the phase 2B test. So this is work looking ahead to the fact when we have the phase 3 trial result and regulatory approval.
Okay, and there's I'm not sure, Brendan, whether you've posted that question 3 times now, and I haven't picked up on it, so I'm doing this now. Brendan to Virginia, how would you propose to test Clostridium perfringens in poultry, as the necrotizing enteritis model is a bit tricky?
Yeah, well, that's the part in which we got the MSD involved. They've been involved in this transgenic in previous projects, and we proposed them like moving that forward, and we discussed the potential which pathogens should we target. They were in this because it's a lot of concern. We decided going for Clostridium perfringens, and they are actually the ones that developing this model because they will have other potential things to test on that. And yeah, we didn't touch. I know the details why it's quite tricky, like all this priming with a fish protein and Emeria maxima to a lot. But yeah, I don't know the details why it's pretty complicated. I think they've done a lot of progress from our meetings and it should be suitable, but I cannot give you much detail because there's not.
You've given me enough. So you went to Merck, is that what you're saying?
Yeah. Yes.
No, well, we basically decided the pathogen we wanted to target.
Yeah. Okay.
Yeah, I mean, we didn't choose ourselves Clostridium and then go to them. We choose together what could be a quite interesting pathogen to target because it's also something infecting in the same area, like local immunity could be more interesting from our areas. So.
Okay, I will sharpen up my Merck contacts for our vaccine.
Oh, that's such a clever phrase. You sharpen up your Merck dome contacts. God, Brendan, you got it nearly all in there. Excellent. Thank you, Virginia, for the answer. So, as we have a couple of minutes left, I will ask one final, potentially slightly biased question, but I would like really all of you to think about it and what's your opinion about it. So we have done in the TB field studies on vaccines in cattle, mainly using the breeds present in Europe, which are the black and white ones you can see running around. And these vaccines do not seem to have the same efficacy when we are actually using them in the cattle which are present in low and medium income countries. And that has really raised an issue that, or the question whether the vaccines we're developing in the so-called first world countries, or say specifically in Europe, using our European genetical material, whether these have any relevance for the situation independent of human side or veterinary side in low and medium income countries. So I guess, Richard, the approach that you choose to test it directly in South Africa is the ideal approach. But is that something we really need to take into concept as well, that we have to develop vaccines suitable for on a European background and suitable on a low and medium income country background, being it South America, being it Sub-Saharan Africa, being it Southeast Asia? And I think this is really something which would affect all of us, because if I look at the genetics of chicken in Southeast Asia and the ones we're using here, there's a lot of differences in there. What's your opinion about that?
Park, would you like to talk to this?
Yeah, so it's interesting you mentioned it. And I think on humans, what we see with the only currently used vaccine, that is BCG, we saw indeed that in the countries where it was developed or where it's mostly used and effective against getting out TB, it worked very well. And then some studies in India and Vietnam found less efficacy from these vaccines. Right. And the hypothesis for this is that it could be due to the strain variation that differs in all of the countries. We have a version of Mycobacterium tuberculosis in, for example, Vietnam, and we have a strain in Tanzania. Those are not the same strains, and efficacy from the vaccine will not be the same. Same accounts for the BCG strain. There's variable efficacy when using the strain that's been developed in Japan, if you use it in one part of the continent as compared to other. So I think this is a very interesting point, and definitely something that should be taken into account.
So we need to take not only differences in pathogen genetic into account, but also in host genetic, which makes it then even more expensive to bring vaccines on the market? Or are we accepting that there will be a cutoff where we say, okay, if we achieve 70% efficacy in one country and 80% in the other country and 60% in a third country, then this is what we have to live with. Brendan, what's your opinion about that?
I think that's what we have to do, and I think that has been done to some extent.
Okay.
I've mentioned the BCG. You know, I think in some countries it's been shown to be really effective. So, similar to, I guess, the coronavirus, where the community immunity, as I call it, rather than herd immunity, there's a figure of 70%. So, I guess you could, use those, get the modelers involved. Have enough installed, and they'll come up with a figure that will be suitable for different populations. I think that's the only really rational approach.
Okay, Virginia, would you support that statement also for the chicken populations?
I think in chickens, the same one you mentioned about using mice, we quite lucky because we can easily adapt the final host. In this way, the chickens are different continents, but it's a quite integrated industry. So what is the intensive production? They actually quite similar in most places.
Okay.
And that way, obviously, but also in the case of, because we're working on the top of the live vaccine, there's also only the big the intensive industry with this expensive vaccine. So usually small producers are not really using the live vaccine. So I think we're quite lucky on that side. It's a bit different. But for example, something we should try to move is that we have tended to do many experiments on inbred lagers, for example. So you're not always using the right model.
Yeah.
Usually for initial trial, it's easier to use chicken that you can keep in cages and the labs, like on the experimental settings for animals, and obviously working with broilers. We doing it now, like especially the Mare Blake, and like it complicates, but yeah. So I think for all this, there's the same problem. Like for strong industry, we can give better vaccines.
Okay.
No, for the small.
Well, I guess the summary sentence and to Phil is that, Phil, we know where you live, and please make sure that you have a variety of money growing trees in the garden so that we can all benefit from that.
Yes, I know that. I have to say that we are working because the comprehensive spending review there was only 1 year funding to tide over because of COVID, and we're looking for 4 to 5 years in the future. And within UKRI, there is a big move to put in a proposal. I'm not saying it is going to win, but there is a strong support for it for a cross-organizational bid on tackling infectious disease, stroke pandemic. So I think the COVID thing has really highlighted that this is important. So I think I am hopeful that more money will come in this area, and more funding will come through some of just the normal calls, because people will begin to realize the importance of these problems.
Okay. Well, if you need any support, now some key players.
Yes.
And we would be more than happy to support anything you would like to put forward to the UKRI.
Yeah.
And I think with that, we have officially 2 minutes left. If there are no other comments or questions coming, there's one more comment from the audience. Correct point from Virginia. We have many vaccines which can be used globally in chicken. absolutely correct. I agree with that. So let's take this as our final comment. I thank all the speakers for providing thought-provoking answers. In part, I think we're agreeing all on things, but there's definitely enough in there where we can collaborate further. It would be nice to see maybe something similar to the current Bloomsbury set happening again in the not so far future. And with that, I hand back to Paul, I guess, or to Ray, or whoever's online.
Yeah, it's me, Dirk. Thank you. So thank you to all of our panel for that really interesting discussion. And I think some fascinating points raised there. And thank you also to the audience for putting in your questions. You probably can't see all one another, but we've had over 70 participants in the session, I believe, from almost every continent, which is really exciting to see. I do have just a couple of slides just to put up. If you've enjoyed this afternoon, then we'd very much like to welcome you to sign up for our next event. We're going to hold a conference the week commencing the 22nd of February. They're going to be different sessions each day, but we won't be doing a sort of 9 a.m. to 5 p.m. day. It will be individual sessions at different times of the day, so that you can dip in, dip out, not a continuous flow of presentations, because as we all know, being on Zoom or any platform for very long periods of time is very wearing. So we're mindful of that. We'll have fairly short, sharp presentations and opportunities for audience interaction. The registration for that event will open at the end of this month. And as I said, the sessions will run each day from the 22nd to the 26th of February. The topic will be broader than today, so we'll be looking at tackling global health and disease challenges through interdisciplinary knowledge exchange and innovation. So we'll include aspects of vaccines, but also looking at diagnostic tools, looking at disease modeling with mathematical tools, and allied aspects in terms of the sort of social and cultural aspects of infectious disease control, and how we can integrate these in a sort of interdisciplinary manner. So please look out for that. We'll be widely advertising that event, as I said, at the end of this month. It will be posted in lots of different places, but you're very welcome to attend. It's an open meeting. Presentations will have been arranged, but there will be opportunities, as I say, for audience interaction. Could I just have my final slide then in that case? Thank you. So I'd just like to reiterate what Dirk said, to thank all of our speakers today. Been really useful discussion, I think, and also to thank my Bloomsbury Set colleagues. You can't see any of them, they're all hiding today behind the screen, but we have a small team at the Royal Veterinary College who are providing the programme support for the Bloomsbury Set. And I'd just like to thank them for all the work they put in to make this session go so successfully today. So thank you everybody for listening, and we hope that you will join us, as I say, in February for our next event. And also we'll be running further symposia later in the spring and early summer. So hopefully you'll keep an eye out for that on the different channels. If you have any questions arising from today's symposium, you want to get in touch with any of the speakers, then please drop me a line, or indeed contact any of my colleagues directly with your questions. If you're interested in collaborating with members of Bloomsbury Set for any of the projects that we're working on, or you'd be interested in commissioning new work, again, we're very open to suggestions. We have submitted a follow-on proposal to the Bloomsbury Set to Research England, who are our main funder, and we're hoping that that will allow us to progress this work from the middle of this year, when the current programme comes to an end. but we're very keen to work with a whole variety of partners. So please, if you're interested in talking to us, get in touch.
And for those of you who enjoyed listening to the talks, why don't you tap us on Twitter and make it known, and maybe we can increase the numbers of participants for the next sessions to come.
That'd be great. Thank you. Okay, thank you, everybody. We're going to close now. Thank you very much for listening, and take care, keep safe. Bye-bye.
Thank you very much, everybody.
Bye.
Bye.
Bye.